Thanks for the introduction. Firstly, I and the rest of our team would like to thank the meeting organizers for providing us the opportunity to share our work today. I'm currently an employee of AstraZeneca. However, all the work that I will present today was conducted during my tenure at the Simmons Cancer Center. The approval of CDK4/6 inhibitors has significantly improved the clinical outcomes of patients with metastatic ER-positive breast cancer. Despite these remarkable initial responses, nearly all patients eventually develop resistance to these agents. Genomic profiling of metastatic breast cancers progressing on CDK4/6 inhibitors has identified several resistance-associated somatic alterations. However, the frequency of these mutations is rather low. Therefore, many novel genomic drivers of CDK4/6 inhibitor resistance remain to be discovered. Accelerated mutagenesis approaches provide a robust and unbiased platform for the discovery of genomic alterations that confer drug resistance without introducing gene dosage or allelic imbalances as frequently noted with traditional approaches. Here is an overview of our screening approach. Deficiency of DNA mismatch repair proteins such as MSH2 results in a high nucleotide substitution rate, which accelerates genomic evolution to allow the accumulation of several genetic alterations, including ones that drive drug resistance. We use CRISPR-Cas9 to delete the MSH2 gene in MCF7 and T47D ER-positive breast cancer cells. DNA repair deficiency generates a heterogeneous population of founder mutants, wherein each founder represents a distinct mutational event. To distinguish between separate founder events, we used a barcoding approach. To barcode the cells, we transduced MSH2 knockout MCF7 and T47D cells with lentiviral barcode library containing approximately 1,000 unique DNA barcodes. Next, barcoded cells were expanded for approximately 25 doublings to allow the accumulation of random mutations. Clones resistant to CDK4/6 inhibitors were selected in the presence of IC90 concentrations of palbociclib or abemaciclib for 4 to 6 weeks. CDK4/6 inhibitor resistant clones with unique barcode IDs were then subjected to whole exome sequencing to identify a spectrum of genomic alterations associated with drug resistance. We generated several MCF7 and T47D clonal lines with biallelic MSH2 loss that failed to express detectable levels of the MSH2 protein. To negate any clonal idiosyncrasies, we screened 8 T47D and 7 MCF7 MSH2 knockout clonal lines. Whole exome sequencing of CDK4/6 inhibitor resistant clones showed increased frequency of mutations ranging from missense to frameshift alterations. Here are the top 10 novel hits identified by our screen. Candidate single nucleotide variants were distilled based on gene mutation frequency across all the resistant clones. Majority of alterations noted in these 10 candidate genes were frameshift or truncating mutations suggesting loss of function. A secondary screen identified loss of ASXL1, ATR, and Mis18BP1 as top 3 hits. Next, in collaboration with Dr. Nikhil Wagle at Dana-Farber, we interrogated the clinical prevalence of these alterations. Whole exome sequencing of 76 tumors that had progressed on CDK4/6 inhibitors identified ASXL1 alterations in 6 patients with either acquired or primary resistance. We noted similar prevalence of ATR alterations in CDK4/6 inhibitor refractory tumors. Analysis of approximately 1,800 post-CDK4/6 inhibitor tumors in the Tempus database also identified several instances of ASXL1, ATR, and Mis18BP1 mutations. Due to the relatively higher prevalence of ASXL1 alterations in CDK4/6 inhibitor-resistant tumors, we focused further analysis on delineating the mechanism through which ASXL1 loss promotes drug resistance. Before we dig deeper into the mechanism of action, let me give you a brief primer on ASXL1 function. Heterozygous truncating mutations in the ASXL1 gene is a common event in myeloid malignancies, where it has been causally linked with oncogenic transformation. ASXL1 inactivation induces epigenetic and transcriptional reprogramming through global loss of the H3K27 trimethylation chromatin repressive marks. While these studies provide evidence of how ASXL1 loss can promote oncogenesis, it is unclear how ASXL1 inactivation overcomes G1S blockade in CDK4/6 inhibitor resistant breast cancer. So we stably transduced T47D ER positive breast cancer cells with control shRNA or shRNAs targeting ASXL1. These shRNAs resulted in about 70% reduction in ASXL1 expression. These are 12-point dose response assays demonstrating that compared to control cells shown in black, ASXL1 deficient cells shown here in maroon and gray were highly resistant to palbo, ribo, and abama. Here are T47D ASXL1 xenografts confirming maintenance of resistance to palbociclib treatment in vivo. In order to delineate the mechanism through which ASXL1 depletion overcomes CDK4/6 blockade, we conducted RNA-seq of SH control and SH-ASXL1 cells treated with vehicle or palbo. Here is a PCA plot showing that palbo treatment induces a dramatic shift in the transcriptional profile of SH control cells, as shown by the clear separation between purple and blue dots shown here. The transcriptional profile of SH-ASXL1 cells, shown in green, was quite different compared to control cells. However, palbo treatment did not induce further changes in their transcriptome. Gene set enrichment analysis showed robust induction of E2F target genes, confirming loss of palbociclib-induced G1S blockade in ASXL1 depleted cells. I had shown earlier that we found several instances of ASXL1 mutations in tumors that had progressed on CDK4/6 inhibitors. In order to confirm whether ASXL1 mutations that were acquired at progression were indeed loss of function alterations, we generated a ASXL1 loss gene signature from the RNA-seq of ASXL1 deficient ER-positive breast cancer cells. Then we queried clinical RNA-seq datasets and found that compared to ASXL1 intact tumors with ASXL1 E513 and E923 truncating mutations showed stronger ASXL1 loss gene expression profile. This is another example from the Metastatic Breast Cancer Project RNA-seq dataset. Analysis of post-progression biopsy showed the presence of ASXL1 RGGE65 frameshift mutation, and it was associated with a stronger ASXL1 loss signature compared to baseline biopsy. Next, we wanted to discover the underlying mechanism through which ASXL1 loss promotes resistance to CDK4/6 inhibitors. We started by looking for any low-hanging fruit. So we interrogated various components of the cell cycle machinery. We took T47D cells stably expressing control shRNA or shRNAs targeting ASXL1 and treated them with palbo, ribo, or abama. As you can see here, CDK4/6 inhibitor treatment resulted in robust suppression of RB phosphorylation in control cells. ASXL1 depletion, on the other hand, led to strong induction of RB phosphorylation, which remained unperturbed in the presence of CDK4/6 inhibitors. We also saw upregulation of CDK6 and 2, overexpression of cyclin E, which is an early E2F target gene, cyclins A and B, and downregulation of endogenous CDK inhibitors such as P21 and P27. To identify potential vulnerabilities of ASXL1 deficient cells, we carried out systematic targeting of various components of the cell cycle that were selectively upregulated in ASXL1 depleted cells. Knockdown of cyclin A led to near complete growth inhibition of ASXL1 depleted cells. In interest of time, I will not be able to show the data, but this vulnerability to cyclin A targeting was not associated with loss of RB phosphorylation. Since cyclin A can partner with CDK1 to regulate G2M transition, we next targeted CDK1. CDK1 depletion phenocopied growth inhibitory effects of cyclin A depletion, thereby presenting a clinically actionable vulnerability of ASXL1 deficient cells. We used RO3306 as a tool compound to test the effectiveness of CDK1 targeting. CDK1 inhibition led to robust growth suppression. Cell cycle analysis of ASXL1 deficient cells, shown in the bottom right panel, showed that SHASXL1 cells remained in a proliferative state upon palbociclib treatment or CDK2 inhibition with K03861. On the other hand, CDK1 inhibitor RO3306 greatly diminished S phase and arrested the cells in the G2M phase of the cell cycle. So in summary, I showed that accelerated mutagenesis screening platform captured a spectrum of novel mutations associated with resistance to CDK4/6 blockade. ASXL1 deficiency leads to robust induction of several cell cycle genes, thereby bypassing G1 arrest induced by cell cycle inhibitors. While CDK2 inhibition had no effect, knockdown of cyclin A or CDK1 led to complete inhibition of the growth of ASXL1 deficient cells. CDK1 may present a clinically actionable vulnerability of ASXL1 deficient CDK4/6 inhibitor resistant cells. With that, I would like to thank my mentor, Dr. Carlos Atiaga, and Samantha Chatterjee, who undertook lion's share of this work. Our bioinformatics team, Ji Woong and Yun Guan, our collaborators at DFCI, Dr. Nikhil Vagle, Jorge and Isha, Alex Barrett at Tempus for providing us clinical data, our funding agencies for supporting the study, and thank you all for your time and attention.